David, will you be?
Okay, good. There we go. All right, should we get started? It says meeting in progress. Okay. Thanks everyone for joining us. My name is Karen Howell. I am scientific advisor and part of the TASCO team for families SCN2A. I just have a couple of announcements before we get started with our guest speakers for today. And the announcement is, please remember to register for our Families SCN2A Summer Conference this year in Anaheim, California. Register by May 31st and don't miss the great scientific and clinical talks that we have prepared for you. The second announcement also has to do with the summer conference. We invite you to submit your abstracts also by May 31st. If you submit an abstract, you are eligible possibly for a poster abstract prize. You're eligible possibly for travel grant. So please look into that, see the website, and submit your abstracts soon. Okay, moving on to today's webinar. It is our pleasure to host the Autism BrainNet and members of the Simons Foundation, who are going to tell us about the amazing resource that they have for scientists in SCN2A. We have David Amaral, Carolyn Hare, Kelly Gleason, and Julia Summer to tell us about the Autism BrainNet and the IPSC repository. So thank you to our speakers for being here, and I will turn the time over to them.
Thank you. I'll go ahead and share my screen and get this going. Okay, well, thanks very much. It's our pleasure to be able to give a presentation on Autism BrainNet as a resource for investigators. We are missing one of our team members, Kelly Gleason, who's our tissue manager, who had a family health emergency that she's trying to take care of today. So she won't be presenting, but the rest of our team are here. So we'll just start with a brief overview of Autism BrainNet. We'll talk a little bit about the Autism BrainNet collection. We will briefly talk about how we process brain donations when they come in, the postmortem brain tissue that's available, some of the clinical data about our donations. We'll then go into the IPSC resources at the Simons Foundation, and then the tissue request process and finalize. So what is Autism BrainNet? Autism BrainNet is a program of the Simons Foundation Autism Research Initiative. We were established in 2014. And we endeavor to build an essential resource for research into the causes of autism and other neurodevelopmental conditions. Thus far, we've been able to acquire 360 postmortem brain donations, and I'll say more about that in a moment. We have a distributed team around the country and, in fact, around the world. So we have 3 collection sites for donations. One is here at the Mind Institute in Sacramento. We have a second that's at UT Southwestern in Dallas, Texas, and a third that's at Mass General Hospital in Boston. We have an affiliate group at Mount Sinai in New York that handles some of our resources. And then we have international nodes. One is at Oxford University in the UK, and one that has recently been established in Canada is the Douglas Bell Canada Brain Bank in Montreal. When tissue comes into any one of these locations, it goes into a consolidated database, and then investigators can obtain tissue from any of these locations where the tissue are stored. We have a group of folks who handle the administration. I want to particularly mention Marta Benedetti, who's the co-director, who's on vacation at the moment. That's why she's not with us. And then the clinical team, Carolyn and Nicole, who you'll hear from Carolyn today. And you've already met Lilian Acosta Sanchez, who's our outreach manager, and Cece McBride, who's an outreach specialist who works with her. So Carolyn was going to talk about our community partners.
Thank you, David. And thank you for having us here today. I really appreciate it. So in addition to the team that David just described, our Autism BrainNet team, our team extends beyond all of those folks to our community partners, including you all at that Families SCN2A Foundation. We rely very heavily on our community partners to understand and be well educated about the importance of brain donation, about the brain donation process, which we'll share with you a little bit more about today. But I hope that in return for our reliance on our partners in the community, that we are providing an important and valuable service, but most importantly, the support that your communities deserve as families become donor families to Autism BrainNet. Now, in terms of the donation process, we work very hard behind the scenes to make this process go as smoothly as possible for our families. The most important thing is to call our 24/7 helpline at 877-333-0999. And the purpose of that phone call is to alert our team that a death has occurred or is imminent when a family has made the decision to donate. We will talk the family through the informed consent process and then get to work behind the scenes to coordinate recovery, make sure that that donation is safely transported and preserved at one of the sites that David just described to you. And finally, our team, Nicole or myself, will follow up with our donor families to learn a whole lot more about the person who's become a donor to our program, and importantly, to provide ongoing support for our families.
Thanks, Carolyn. And just want to emphasize one final detail about this is that there is no cost to families for brain donation, and it does not interfere with autopsy and funeral plans. So the donor criteria, individuals with a diagnosis or suspected diagnosis of autism, there are no exclusions. We are accepting donations from individuals who have other related health conditions, both genetic and idiopathic forms of autism. We also, and this is where SCN2A comes into play in a sense, that individuals with a diagnosis of autism, without a diagnosis of autism, sorry, but with a genetic diagnosis linked to autism, are acceptable donations as well. And the list of genes is found at sparkforautism.org. And then finally, we do also accept donations of individuals with no known serious neurological or psychiatric condition as comparisons for those with autism or genetic conditions. And all of these donor criteria are on our website. We'll show you that in a few minutes. So this is our collection at the moment. We have 366 donations, In green are the donations that are individuals who have a confirmed diagnosis of autism. And I should say that both Carolyn and Nicole, as she mentioned, after the donation is completed, carries out an interview process with the next of kin. Part of that is to determine or confirm, in fact, that the individual did have a diagnosis of autism, but we try and collect as much phenotypic information as possible. So while there's 134 cases that have been confirmed, there are additional cases, 19 currently, that are awaiting that confirmation process. And you can see that we have a fairly large sample of control cases. And the idea is when we distribute tissue to investigators, we provide them with not only the case material, but also a control that is as closely matched in terms of sex and age and postmortem interval as possible. This shows the postmortem interval for a collection of cases, and what it emphasizes is that we try and bring the donation into one of our collection sites for further processing as soon as possible. Obviously the quality of the tissue for research is optimal the sooner it comes to one of our sites for processing. So we aim for 24 hours or less, although we do allow donations that exceed this in certain situations. Since autism is more predominant in males than females, our collection donations thus far sort of reflect that. So we have more male donors than female donors. For SCN2A donations at the moment, we have 3 donations, and these donations have been with an average age of around 9 years. That's average of all 3 donations, and you can see that what led to the donation is death due to either epilepsy or other issues that did not affect brain quality. The processing, half fixed, half frozen, I'll explain that in a moment a little bit more, but we try and process the donations in a way that is optimally useful for all kinds of scientific investigations. So half of the brain is fixed. Usually the fixed tissue is used more for histological and neuropathological studies. The frozen tissue is used more primarily for genetic studies. So this is what I was referring to. We've actually adopted a process now where we minimally fix the brain. So the brain is only fixed for 36 to 48 hours. That allows optimal tissue preservation, but also quality for scientific investigation. That tissue, after the fixation process, is cryoprotected and frozen. And that tissue then is really useful for decades after it's been processed. The other hemisphere is sectioned and flash frozen without any fixation. And again, that tissue is optimal for genetic studies. We do have some tissue that was processed in different ways earlier, and I should say that we inherited, when Autism BrainNet started, the tissue that was collected by the Autism Tissue Program, a program of Autism Speaks, and much of that was fixed in a different way. So here's, again, just a different look at our collection at this point. So of the autism cases, for example, we have 70 that are frozen that have been confirmed, 43 that have been frozen but are under review. Here's the fixed tissue, but longer term fixed tissue. And here's the fixed tissue that is using this minimally fixed tissue that we implemented just a couple of years ago. So now I'll turn it back over to Carolyn, who'll run through the clinical data that are available through for Autism BrainNet donors.
So as you're having an opportunity or taking a moment to read through some of the clinical data available via Safari Base, what I would really like to emphasize is that any investigator with any clinical query that they can reach out to us at any point, and we're happy to provide information that, of course, is readily available in Safari Base, but may not be readily available in Safari Base. So, for example, just last week, we had a researcher interested in Prader-Willi syndrome who was wanting the BMI or the height weight for each of our donors. That information was not presented in Safari Base, but something that we can easily put together and provide to the researchers. And so I hope that as you look into this resource, if there's something that you're interested in that you don't see readily available, that you simply reach out to us so that we can find that information and provide it to you.
Yeah, good. Thanks, Carolyn. I do want to just emphasize a couple of things here, that all donations that come into Autism BrainNet undergo genomic characterization. So we're all doing whole genome sequencing, whole exome sequencing, and arrays. And one thing that would be important concern for coroners and medical examiners is that if they are willing to release a donation, a brain donation, but they also need a neuropathology report. We have a board certified neuropathologist on our staff who will provide a certified neuropathology report, and that's free of charge. So it's actually a savings to medical examiners, which are facing tight budgets often. So I'm going to turn it now over to Julia, who's going to talk about the IPSC resource availability. Julia.
Hey, everybody. Thank you so much for giving me the opportunity to talk a little bit about the induced pluripotent stem cell resource we're making available. I'm going to jump right in and talk about what specifically we have for the SCN2A community. So at the moment, we have induced pluripotent stem cells available from 12 individuals with 12 different variants in SCN2A. And those are available for request by any researcher, academic, or for profit. We are also in the process of making iPSCs from 3 more individuals with 3 additional variants. And those are almost ready to be requested. They're currently undergoing QC. And anybody who would like to have a more careful look at what exactly we have, please scan the QR code, and this will lead you to a Google sheet that in detail lists the variants that are represented in the iPSC resource. And you'll see we have also iPSCs for many other genes, neurodevelopmental disorder genes linked to autism. You will see that the variants represented for SCN2A include splice site, predicted loss of function, and missense mutations. We will also soon have, for 3 of the SCN2A individuals, iPSCs from sex-matched familial controls available, and more may be added. And this is something we're trying to match control creation to some degree to the use of the cells. So if investigators are interested in using the cell lines of a particular donor and they want a familial control, they should reach out to us, and we can discuss if we can make that control if we have blood samples from those familial controls available. I also wanted to point out, because this is a question we get a lot, that we are not systematically creating CRISPR-corrected isogenic controls. However, similarly here, if investigators are working with a particular cell line and they are in need of isogenic control cell lines for that, they should reach out and we can discuss if there is a way to create those control lines in collaboration. What has happened so far is that often investigators to decide to create those CRISPR corrections themselves, maybe with a stem cell core at their university. And they then share those control cell lines back with Safari so that we can broadly distribute them to the community. We can go to the next slide. So just to give a little bit of an overview where those iPSCs come from. So the blood is initially collected through the Simon Searchlight study. So participation in Simon Searchlight enables participants to donate blood. And as you know, the Simon Searchlight gene list is long and includes 150 plus genes linked to autism. Once we have a good number of blood samples, we review the variants carried by those donors. And I would say the utmost important criterion to make iPSCs from a donor is that the donor is actively engaged in Simon Searchlight, so also participating in contributing phenotypic information. And this is because we believe that this resource becomes particularly strong due to its link to the phenotypic information collected on individuals. Another criterion is investigator interest. We try to create iPSCs from individuals with variants that we know will be worked on. And if there's investigators who particularly want to work with a specific variant and we happen to have a donation for that variant, that would be a strong reason to make iPSCs for that individual. And otherwise, we try to base selection on scientific criteria, basically judging how likely the variant is truly causal for this individual's disorder. So those would be splice site loss function mutations. Then we go down to recurrent missense, and the lowest priority would be non-recurrent missense mutations. The blood is shipped to Sampled, the biorepository we work with, and they extract the PBMCs. And the reprogramming to iPSCs actually happens at the New York Stem Cell Foundation, and they use a Sendai virus delivery for the reprogramming factors. In most cases, we're able to get 2 clones. We only distribute clones that pass all these QC measures listed here on the bottom, so it's pretty rigorous. We can move on to the next slide. So all the iPSCs, as well as any genetic information we may have, phenotypic data, is available for requests through Safari Base. This here is the URL you can use to access Safari Base. The steps to request iPSCs are you need to create a Safari Base account. You need to sign or join an existing researcher distribution agreement. And then you create a project. Project also includes providing us with either an IRB approval or exemption letter. And then you submit a request for biospecimens, in this case, iPSCs. And for this request, you will need IDs. And these so-called Safari IDs are listed on that Google sheet that I linked to before, but I'll also post it here in the chat one more time. And so you can now specify which exact cell lines you would like. The iPSC distribution happens through the biorepository Sampled, and Sampled is also the one that arranges for the shipping. Currently, iPSCs cost $120 per vial, which is, I think, a lot less than from many other places that distribute iPSCs. And the investigator, however, has to cover the shipping costs. And if you have any questions about the request process or ordering or shipping, it's always best to reach out to sdbr@simonsfoundation.org. And if you have any more questions about QC or about variant availability or any other science questions about the IPSC resource, you can reach out to me, which is jsommer@simonsfoundation.org. Thank you.
Thank you, Julia. Okay, so as Julia said, there's a process for doing tissue requests, and this is the part of the program that Kelly would have presented to you. But just to talk now specifically about the tissue request for the postmortem brain tissue. So approved researchers worldwide can apply to receive access to Autism BrainNet, the inventory and tissue via Safari base, just as Julia had mentioned. The same process takes place where you have to establish an account and do the various steps that Julia mentioned. Requests for tissue are vetted by a peer-reviewed scientific committee for rigor and relevance. And then in all cases, Safari data sharing policies require that investigators pledge to deposit data from their research back to Autism BrainNet to enrich the resource for future studies. The tissue and information that goes out to investigators are all de-identified. Kelly can be approached for additional information about what tissue is available or how it's distributed at request@autismbrainnet.org. She handles that account. Just to again reiterate that researchers interested in accessing Autism BrainNet data can review the full tissue catalog on Safari base, and again the request for additional information can go to request@autismbrainnet.org. One difference in terms of the cost for tissue is at the moment there are no costs to the investigator for the actual tissue. However, just as for the IPSCs, the investigator does need to cover the cost of shipping. And we have already done a number of distributions and had distributions worldwide, as far away as Australia and Italy and Spain, and we manage all of that shipping in coordination with the investigator that has applied for the tissue. And with that, I think, Carolyn, you want to just finish up?
Sure. I will finish up by saying thank you again for the opportunity to join the Families SCN2A Foundation today to share a little bit more about the bioresources available through the Simons Foundation. There's a lot of work that goes into this behind the scenes, but I hope that we've made this work meaningful and relevant to the science community and that we hear from you so that we can continue to share this important resource.
Good. And thank you from us to the whole team to you as well for being a partner. So I'm going to go ahead and stop sharing at this point. And we're happy to answer any questions if there are any.
We thank you so much, David, Carolyn, and Julia. That was very clear and helpful.
Good. Welcome.
And of course, if you hear any questions, you can always pass them along to us or connect us with people.
Absolutely.
Absolutely.